So good afternoon, everybody. Thank you for coming to our weekly departmental seminar. It's my great pleasure to introduce Joe Pickrell. So I think I first became aware of Joe's work back in the, it's been quite a few years now, back when he and others were posting on a Genomes Unzipped. There's a blog still out there on the web. It's been a couple of years, I think, since you guys have posted on there, but
Yep.
There's a really nice blog about different elements of genomics, and which is, of course, an interest of mine. Joe did his PhD with Jonathan Pritchard. Was Yoav also coming, or was it just Jonathan? Were you and Yoav too? Was he
just Jonathan.
Just Jonathan. Okay.
Yep.
So he actually did some really excellent work linking genetic variation with gene expression before moving to Harvard and working with David Reich with population admixture and population studies. Now I think today he's going to tell us about some more cool human genome stuff that he's been working on, and yeah, really excited to hear about it.
Thanks.
Take us away.
Thanks a lot. Thanks very much for having me. So again, my name's Joe Pickrell from the New York Genome Center. And I titled my talk today, What Can We Learn from Human Genomics at Scale, and how do we get there? And so I'll come back to this question a few times during the talk. And it's really an absolutely fantastic pleasure to be here at Michigan. I've said publicly that I'm a huge fan of everything that's going on here. Like, the Michigan imputation server has saved me over and over and over again, probably like thousands of dollars. So anyone out there who works on that, thank you very much. It's an amazing resource. And so what I am interested in, and what I think probably many of you are interested in, is what the fundamental question in human genomics, which is which genes or genotypes influence which phenotypes, and how do they do it? And this is basically genetics. So how does genetics work, right? And so as I started my lab a few years ago, we sat down and we thought, how can we make progress on a question like this? And it's actually really challenging to decide what to do. So on the one hand, there's huge value in improved genomics methods. People have gone from doing genetics using blood groups to microsatellites, to tag SNPs, to whole exomes and whole genomes. And there's been a lot of the progress in genomics is progress in technology. And so there'd be huge value in putting effort into developing the next generation of technologies for measuring genotypes. There's also huge value on the other end. So and there's huge value in the phenotype side. So if you can connect genotypes to phenotypes, you need great methods for measuring phenotypes. And so there's a lot of great work on extracting phenotypes from electronic medical records, on deciding which phenotypes matter for which disease and measuring those instead, and then sort of using the mobile technologies to extract entirely new classes of phenotypes and connecting those to disease. And so there's a huge value in spending time on the genotype side. There's huge value spending time on the phenotype side. There's also a lot in this arrow. So what's in the arrow? There's huge value in doing statistical modeling, improving the way we map genotypes to phenotypes. There's huge value in EQTL mapping and in other functional genomics things. And so actually picking apart this arrow, which is totally simplistic, there's a lot of really complex and interesting biology in between genotype and phenotype. And so this gives you a lot to think about, right? So how would you spend your time? And so we spent a lot of time thinking about how should we spend our time in this problem. And I started reading things outside of genetics. And so I want to briefly go over a paper that's been fairly influential, which is actually from natural language processing, things like machine translation. And this is from a few authors at Google who, and they titled their paper, "The Unreasonable Effectiveness of Data," which is a nod to an earlier paper on the unreasonable effectiveness of mathematics and physical sciences. And so they argued that what's been unreasonably effective in natural language processing, machine translation, is data. And they took a few lessons away from this. One is that more data beats better data. So I'll go ahead and just read this. So one of us, as an undergraduate at Brown University, remembers the excitement of having access to the Brown corpus, containing 1 million English words. Since then, our field has seen several notable corpora that are about 100 times larger. In 2006, Google released a trillion-word corpus with frequency counts for all sequences up to 5 words long. In some ways, this corpus is a step backwards. It's taken from unfiltered web pages and contains incomplete sentences, spelling errors, grammatical errors, and all sorts of other errors. It's not annotated with carefully hand-corrected parts of speech tags. Just the fact that it's 1 million times larger outweighs all these drawbacks. And so in this particular field, if you had a trade-off between collecting more data with a bunch of errors and poor annotations versus really well annotated data, you would choose more data. So more data in this field beats better data. And likewise, more data beats better algorithms. And so again, quoting from this paper, simple n-gram models or linear classifiers based on millions of specific features perform better than elaborate models that try to discover general rules. In many cases, there appears to be a threshold of sufficient data. And so again, in this application, taking simple statistical models and applying them to more data is a better use of time and effort than developing more sophisticated statistical models to apply to less data. And so this led us to a thesis. And I'm not going to say this is the way the world works. This is just an operating thesis. So in genetics, so we know how to measure genotypes, and there's a lot of value in coming up with better ways of measuring genotypes. We know how to measure phenotypes, and there's a lot of value in coming up with better ways of measuring phenotypes. And there's a lot of value in figuring out how to connect them, both at the functional level and at the statistical level. But so here's our thesis. So that there's major potential for progress by focusing on scale rather than technology and methods per se. So what I'm going to argue is that there's value, huge value potentially, in just doing what we know how to do, just more of it. And so how does that affect our work? And so we've taken a few lessons from this type of thesis. There are a few implications from this thesis. One, so we try to focus on problems where it's possible to have measurements on many people, large amounts of data, regardless of the precision of those measurements. So we're happy to take poor measurements on 1 million people versus precise measurements on hundreds of people. We try to use the simplest, effective statistical tools for the data at hand. If there's no simple, effective tool, we try to develop it. But if there already is one, let's just use it out of the box. And this led us to the rather, I'd say, unfortunate conclusion that we need to collect some data ourselves. And this was scary for us to just sort of confront and realize that this is what we needed to do, because I'm a statistical geneticist with a 5-person lab and no wet lab. So how are we going to do this? And I'll come to this at the end. And so I'm not saying this is the way the world works. This is sort of our operating thesis right now. And so I'll tell you about a few projects that have come out of this sort of approach to human genetics. The first is a project called Case Control Association Mapping Without Cases. The second is on identifying genetic variants that influence viability. And the third, as I told you, we finally realized we have to collect data ourselves, and this is how we're doing it. And so I'll start with the first project here. And this is a collaboration between Jimmy Liu in my lab and Yaniv Erlich. And there's a preprint on it. Actually, this is published now. And so we started from case control association studies. I don't have to tell people in this room how case control association studies work. You get disease cases, you get disease controls, you measure some genetic variants, and you look for differences. And successful applications of this approach require some pretty large sample sizes. A big study of schizophrenia had about 150,000 people or so, found about 100 genetic associations. But this is sometimes actually really hard to do. Sometimes it's actually really, really challenging to collect disease cases. In a few particular cases, this is particularly notable. Early onset diseases, So there's a big consortium that's trying to collect 50,000 individuals with autism. And this requires a team of people across 21 universities, numerous autism community organizations across the entire United States to get to 50,000. And that alone might not be enough. Late onset diseases like Alzheimer's disease, it's also challenging to get cases for these types of diseases. Or rapidly lethal diseases. This is glioblastoma. Glioblastoma kills on average within a few months. And so getting people into your studies, if you want to do genetics of this, is quite challenging. But as we know, obtaining DNA from relatively healthy populations is easier. So 23andMe, this company, has over 1 million participants. Probably very few autism cases, probably very few Alzheimer's cases, probably very few glioblastoma cases. But it's easier to get 1 million people. Or the UK Biobank, which I'm going to tell you about, our work with that, has about 500,000 participants. Again, probably not a lot of people with these diseases, but it's easier to get DNA from relatively, quote unquote, healthy people. And so is there a way around this need for disease cases? So this is the problem where we're trying to connect genotypes to phenotypes. How can we get genetic data from people that have a disease without measuring their genetics? And so the idea we're going to operate on is what we call genome-wide association studies by proxy. You share 50% of your genome with your parents. So if your parent had a disease, an unmeasured person here, and they had a phenotype, and we were able to measure your genotype and the phenotype of your parent, this is actually like a causal graph here. Your genotype is partially inherited from your parents. And if the genotype is associated with your parent's disease, this induces a statistical association between your genotype and your parental phenotype. And so this is how we're going to do it. Instead of, in order to get the DNA of people who have a disease, we're going to get the DNA from their relatives instead. And so instead of cases, find relatives of cases. And this is an approach used in longevity studies where the cases are long-lived people. Once you've measured somebody's age of death, it's difficult to get DNA from them. Just to sort of drive this home, this is a genome-wide association study by proxies. Imagine you have a genetic variant that influences a disease, and the allele frequency in cases is F sub A. And the allele frequency in controls is F sub U. The allele frequency in proxy cases, which is what we're going to call them, first-degree relatives of the cases, is halfway in between there, right? And so that, with enough people, is significantly different from that. And so, again, proxy cases here are individuals with a single affected parent. Later, it'll be any individuals with any number of affected first-degree relatives. So, of course, this is different from that, but it's also less different than that. So how many more people do you need? And I'll just go ahead and tell you how many more samples do you need if you're going to do this type of approach. The answer is 4-ish. So here's a plot showing the odds ratio of a particular genetic variant versus the log of the sample size necessary to detect that variant at genome-wide significance. It has some allele frequency. If you do a standard case control study, it's the dotted line. If you do in a proxy case control association study in the solid line. And the difference here across odds ratios and allele frequencies is about 4. So you need 4 times as many people. Is that actually valuable or feasible in practice at all? So now I'm going to turn to the UK Biobank. And so the UK Biobank, data field 20107 is illnesses of father. And so all 500,000 people were asked, did your father ever suffer from heart disease, stroke, high blood pressure, chronic bronchitis, Alzheimer's, diabetes, Parkinson's disease, severe depression, lung cancer, bowel cancer, prostate cancer? And there are 150,000 genotyped individuals in the set that we looked at. I should, again, make this point that we're taking, these are very imprecise phenotype measurements. This is diabetes. They didn't even ask the difference between type 1 and type 2 diabetes, just diabetes. And heart disease, what does that even really mean? And And so these are very imprecise phenotype measurements. But there's a potential gain in just that there are a lot of people. So just to really drive this home, in the UK Biobank, there are exactly 15 people, when we looked, that in their medical records had been diagnosed with Alzheimer's disease. And if you want to do a genome-wide association study with 15 cases, you're not going to have a good time. But there are 15,000 proxy cases. So that gives you some power to do something. And basically across the phenotypes that were measured in the UK Biobank, this association by proxy was more powerful than a standard genome-wide association study. So here I'm showing you an effective number of cases and controls corrected for the effect size differences for all of these diseases. In red is the effective number of cases and controls in using this proxy association study. And in blue is a case control study with the case status taken from the electronic medical records. And across the board, the red bar is higher than the blue bar, except for high blood pressure. So in general, in this cohort, ascertained in this way, association study by proxy is much more powerful. And so does it actually work? Here's a Manhattan plot of Alzheimer's disease. Did your parents suffer from Alzheimer's disease? You've seen the Manhattan plot before, but just drive it home. On the x-axis is position along the chromosome. Y-axis is negative log 10 p-value. You see one peak here that goes up, way up there. This is APOE. So obviously, APOE, the well-known Alzheimer's variant. We also looked at things like breast cancer. It's not just things where there's a single well-known association. This is a genome-wide association, a GWAX, genome-wide association by proxy. Breast cancer identifies FGFR2, which is the top genome-wide association study hit for breast cancer, and two other well-known hits are up there. But this isn't identifying new things, right? So we asked, can we then go ahead and meta-analyze these results with a sort of traditional genome-wide association study? The answer is yes. So the odds ratio from this proxy case control association study is about half as much as it is adjusted for a real. If you took the odds ratio from a proxy case control association study and mapped it onto what it would be if it were a real case control association study, it depends on the allele frequency and the effect size. But overall, you can see on a log scale, it's about 2. So it's about the effect size is about half as much. And so we can extrapolate to the implied odds ratio in a standard case control design. And then we wanted to test, you can think of all sorts of reasons why this sort of mapping would not actually work. So we wanted to compare the odds ratios at known SNPs. So here I'm showing you each point here is a known association study for known association for type 2 diabetes. And on the x-axis is the log odds ratio from the previously reported sort of known association studies. And the y-axis is the adjusted log odds ratio from a genome-wide association study by proxy done in the UK Biobank. Again, on the phenotype diabetes, not type 2 diabetes. Well, you can see each point here, they line up pretty well. The slope is about 0.92. The effect sizes are pretty comparable. But that's not totally always the case. So here's that exact same plot for coronary artery disease. On the x-axis is the log odds ratio for a genome-wide association study of coronary artery disease. And on the y-axis is this adjusted log odds ratio from a proxy association study for quote-unquote heart disease. And again, it lines up reasonably well. But the slope's a bit lower. You can see there's we're somewhat underestimating the effect sizes here. This is probably just because heart disease and coronary artery disease are not quite the same thing, or people are remembering the wrong phenotype. Like I said, there are a ton of other biases here. So some are due to how well people remember the phenotypes. It turns out across the board, females are more likely to report a relative with a disease than males. There's a recall bias here that is pretty dramatic. So overall, the p-value for differences for whether the person in the UK Biobank, females, report a parent with Alzheimer's disease, there's a dramatic recall bias here in the way that women report more diseases of their parents than men. Let's just go ahead and do it. We can go ahead and run our meta-analysis, and here's what it looks like for Alzheimer's disease. We're able to identify 4 new associations for Alzheimer's disease. Some of these are kind of interesting ones in immune signaling pathways. And so in general, we think this works. Well, I removed the other ones, but we find a bunch of other things. So we identify a number of novel loci. So we identified 4 novel loci for Alzheimer's disease, 8 for heart disease, 5 in type 2 diabetes, none for depression. We were unable to analyze the cancer phenotypes because the summary statistics aren't available. That's the only reason why. But we think we probably could have found some things. So in general, when people are collecting cohorts, we strongly recommend you just collect. It's a relatively simple question to ask. Ask about the diseases of parents, of siblings, of children, if there are any. And it's an extremely powerful question to ask to incorporate into studies like this. So that is, we call that case control association mapping without cases. And so in the next project, I want to talk about identifying genetic variants that influence viability, so sort of lifespan associations. And this is a project in collaboration with Molly Shavorsky up at Columbia. And we're using the UK Biobank and the Kaiser Permanente cohort in California. And there's a preprint posted on bioRxiv identifying genetic variants that influence viability in large cohorts if you want to read more. And I want to motivate this with a somewhat different approach, which is the population genetics motivation, which is that methods to detect genetic adaptation work on different timescales. So if we have people living in different parts of the world today, related to sort of an outgroup out here, there have been a number of adaptive changes in human evolution at different timescales. And there are a number of methods to detect adaptive evolution. So things like DNDS, you might have heard of, proportion of functional changes, operate on a really long time scale. And so people have identified immune genes that have more non-synonymous changes than you'd expect by chance over the course of millions of years, which indicates evolution of immunity over time. And on shorter time scales, say tens of thousands of years, you can look for very large differences in allele frequency between different populations. And what we were interested in was how far can we push this? Can we detect genetic changes in populations over the time scale of contemporary populations, say hundreds of years? Is natural selection operating on that time scale? And there's some evidence that this is the case. So there have been a number of population studies, for example, showing that taller men have more children. This is a study from about 17 years ago, looking at men in different ages and comparing the heights of men with and without children. And in general, you can see men that have at least one child are slightly taller. This implies, in principle, that genetic variants associated with, for example, height could be changing in frequency over really short timescales, perhaps over the course of tens or hundreds of years, is how quickly these things could be changing. And so we wanted to ask the question, do genetic variants or sets of genetic variants that influence a phenotype change in frequency across individuals of different ages? And if this were the case, it would imply effects on lifespan or fertility. Either these genetic variants influence how long people live, or they influence how many children these people have. And I'll just go ahead and tell you that in general, what we think we're looking at are effects on lifespan. And so what's the data that goes into this? So first is the Kaiser Permanente cohort, about 80,000 genotyped individuals, 60,000 of European ancestry in Northern California. The UK Biobank, 150,000 genotyped individuals in the UK. And the basic statistical model here or the intuition is that they're people of different ages, right? So here is the Kaiser Permanente cohort. Some people are as young as 21. Some people are over the age of 83, and this is just the distribution of how often you see people of different ages. The average, the median here is somewhere around 61, 66. And they report these ages in bins, bins of 5 years. And so we developed a fairly simple method to just test whether genetic variants differ in frequency across age bins. So here's, you can imagine the genotype of some individual. It's a binomial sample of 2 from some allele frequency. We model this allele frequency with a logistic style regression where, I should show you what those things are actually. The effect of being in an age bin, the effect of principal components to account for population structure, and additional stuff to account for genotyping batch, age by sex interactions. And so you can then predict the genotype of somebody based on all of these things. And there is variation across age bins. Some of it is not particularly interesting. So this is a genetic variant near the gene lactase. And I'm showing you the allele frequency of this genetic variant in the dark colors here in these different age bins in the Kaiser Permanente cohort. You can see there's a pretty significant change in the allele frequency from 42% to 38%. But actually, this is pretty well accounted for just by a model that doesn't include age. So this is our expected line from a model just based on the principal components. So just population structure varies across ages as well. In Northern California, younger individuals have more Southern European ancestry. That's the explanation here. But so we can account for that. And then we can go across the whole genome and ask where are there genetic variants that do depend on age. And here is our Manhattan plot. And this is going to look very similar to a Manhattan plot I showed you earlier because there's exactly one strong association, and this tags the APOE4 allele. So here is in the dotted line is the expected APOE4 allele frequency under a model that accounts for population structure and a bunch of batch effects. In the dark blue is the actual frequency of the APOE4 allele in people with different ages. And what I'm showing you in orange here is the average age of onset of Alzheimer's disease in people with different numbers of this allele. So you have 0 copies of APOE4. Average age of onset is around 85. 1 copy of APOE4 around 75. 2 copies of APOE4 around 68. And so you can see quite strikingly, even in this population, where all we're measuring is their age. We don't know anything. Well, maybe Kaiser does, but we don't know anything about their Alzheimer's or memory or any other medical information about them. We're just using their age. We can see that people with an APOE4 allele, they start disappearing out of the population either due to incapacitation or death around this time. And I'm one of these people here with 1 copy of APOE4. It's a bit of a depressing plot to look at. But we can then go ahead. So that's the one single genetic variant that shows this pattern. We can ask about sets of genetic variants. So now instead of modeling someone's genotype, we can look at their polygenic score for some phenotype, which is just the sum of the effect sizes of a bunch of variants on the phenotype. And then we can plug that into the exact same model and ask whether polygenic scores for different phenotypes vary across time. And we tested a bunch of phenotypes in this. And in the Kaiser Permanente cohort, we found basically nothing. So here is a QQ plot of all the different phenotypes. The expected p-value in the negative log term, the observed p-value for all of the different phenotypes, and we find basically nothing. The strongest signal, so we excluded Alzheimer's disease because of APOE4. It's for agent menarche, and I'll come back to that. So okay, we can now turn to the UK Biobank and see if we replicate these signals or whether there's anything else more interesting. And so there's a really restricted age range in the UK Biobank. It's people that were born were about 50 to 60 at the time then. So we're going to consider instead the ages of parental death. So these people were also asked, at what age did your mother die if she died? At what age did your father die if he died? And so it's sort of like our previous analysis of the proxy association study. And that's how we're going to do this in the UK Biobank. We can do the exact same analysis. And it turns out, so if you look at the father's phenotype, we see one fairly strong hit. So these are individual loci associated with paternal age at death, CHRNA3. We can plot this thing out. It looks like this genetic variant starts declining around the age of 70, 75 or so. So people with this genetic variant start dropping out of the population at around the age of 70, 75. And this genetic variant has been seen before in other genome-wide association studies. It's associated to smoking behavior. So people with this genetic variant, if they smoke, they tend to smoke more. So CHRNA3 is a nicotine receptor, and so people with this genetic variant tend to smoke a bit more if they smoke than people without this genetic variant. And so that suggests a natural hypothesis for what's going on here. We also looked at these polygenic scores. And in contrast to the Kaiser Permanente cohort, we found a number of polygenic scores that are associated with age at death. And so this is for the father's age at death. We find a number of very strong associations led by total cholesterol, coronary artery disease, age at menarche. Note this is in men. LDL cholesterol, BMI is in there. And for mother's age at death, we find a couple of hits. The top one here is age at menarche. This is age at first birth. And so if we look at what exactly these signals are showing, this is the polygenic score. So a later age at menarche is higher up here, and maternal age at death on the x-axis. So people with a genetic score that predisposes them to an earlier age at menarche have an earlier age of death. So that's why the polygenic score is going up here. People with a low polygenic score are dropping out of the population, right? This is potentially consistent with these long-standing hypotheses about trade-offs between fertility and longevity. This is one of the hypothesized reasons for the drivers of the evolution of aging, in that you can divert energy to reproducing early, but with the trade-off that then you don't live as long. And this is potentially the first evidence in humans that this might actually be relevant. And so to summarize this, we can sort of contrast the genetics of lifespan in late century California and mid-century UK. So again, in California, we saw a strong effect of APOE on longevity in both men and women, and little effect of polygenic traits. In the UK, the parents of the people in the UK Biobank, so these are people that were alive around the 1950s, 1960s in the UK, we see this strong effect of CHRNA3 in men, and genetic scores for increased cholesterol and heart disease, and these fertility phenotypes in women. And so what's the difference here? So one possibility is sort of the trivial one, that we're not measuring the same things, and the genotyping arrays are different, but it's definitely not the genotyping arrays. It could be that we're measuring slightly different things. But we think probably what's going on here are things like gene-environment interactions. And the CHRNA3 locus suggests one specific environmental factor, namely smoking. So in the UK in the 1950s, the average man smoked about half a pack a day, which is not the case. More than half of women did not smoke. And so that suggests one potential mechanism for, one, the difference between males and females at this particular locus in the UK, and also a difference with California today. In California today, the rates of smoking are about an order of magnitude lower. There may be other sorts of environmental factors which we're not totally aware of yet that are modifying the genetic architecture of longevity across these two cohorts. And so finally, I want to close with just a few slides on what we've been spending a lot of our time on recently. which is a different approach to inexpensive and scalable genomics. And this has been led by two people in my lab, Tomas Barusa and Kaya Wasik. And we decided after seeing all the successes of these great data that we needed to collect our own data sets. The UK Biobank, what if they didn't ask a question that we were interested in? Or in Kaiser, what if the medical records aren't made available to us? We need to be able to actually do this on our own. So we need to be able to collect genotypes, and we need to be able to collect phenotypes. But there's a problem that I sort of alluded to earlier. This has to be feasible for a lab of, well, so this is being run by two people with no dedicated budget. So that's a serious problem. So what we came up with is a project called Seeq. You can sign up, Seeq.io. It's an app you download, and you can and how it works is we give you your ancestry, microbiome, and ask people questions. And so for $50, it sort of covers our costs. We ship participants saliva collection kit in the prepaid mailer. It comes into our lab, and we set up a very highly automated pipeline, mostly robotics. This is our Agilent Bravo here, for doing all the DNA extraction and library prep. And then we pull the samples for ultra-low coverage sequencing. So we're doing 0.1 to 0.5x coverage sequencing, which seems low, seems very low. And it is low, but it works for most of the things you want to do. So things like ancestry and microbiome, we can get out of that. We can do imputation to fill in all the missing stuff. And then we give people this information back. So ancestry, microbiome, and we ask people questions. And I'll get to that in a second. So for ancestry, what we've implemented is a supervised structure model. So basically, we have a reference panel of people of known ancestry, and then we take you, and if we have 0.1x coverage of your genome, that's about 8 million SNPs. We can take those 8 million SNPs and in a structure model sort of fit you into these sorts of clusters. And this is me here. This is about the same as what I got from 23andMe or Ancestry.com. For the microbiome, we take everything that's not human, we chuck it, and we run everything that's not human through Kraken, which is this great tool by Wood and Salzberg. When we run it against the NCBI NT database, and we summarize it at a very high phylogenetic level. And so here I'm 43% of the bacteria. We also often get, so we get some viruses, DNA viruses that are in there, and food that people have eaten, and whatever's in their saliva. And sometimes it's food, sometimes it's people have asked us, like, why is there dog in my DNA? I'm like, well, do you live with a dog? Yes. Well, that's in your mouth too. And so we can sort of get that information out. And then we ask people questionnaires, and some of this is more serious stuff. So you can see this is sort of like an allusion to what we were working on before. Have you or any of your close relatives, parents, grandparents, siblings ever been diagnosed with dementia or Alzheimer's disease? About 32% of people have said yes. So we can start doing genome-wide association studies by proxy for Alzheimer's disease. Do you have more than 2 nipples? 2% of people say yes. So we can. It's a mix of these sort of amusing and scientifically interesting questions, and it sort of engages people. And so overall, we've implemented the logistics of recruiting participants, shipping and receiving saliva collection kits. This is actually more pain than you can possibly imagine. Molecular biology for inexpensive and automated DNA extraction library prep. And then pipelines for ancestry analysis, microbiome analysis, and imputation from ultra-low coverage data. And one thing we said from the beginning is we don't want this to be our own sort of silo of things. We want people to be able to take their data with them and participate in other projects. So for you don't have to do this from scratch. Like we opened up an API. Here's a Python example of how to use the API. Just import Seeq. And you can create a website that uses all of our infrastructure. So a participant will click on your website. If they're already a Seeq participant, you get their data. If they're not a Seeq participant, We can ship them a saliva collection kit, do all the lab processing, sequence them, and then ship you the data. And so people can now run low coverage sequencing studies, potentially at very large scale, with no lab and no real infrastructure. And so why we think this is important is this now enables genomics on a budget, like a real budget, not a lot of money, driven by small groups who have interesting questions they want to answer, and participants themselves. And so to summarize what I told you, first, project and case control association mapping without cases, where you can replace disease cases with family members to run large studies. Second, identifying genetic variants that influence viability. And I showed you this example of potential gene-environment interactions between California and the UK. And finally, I briefly described the approach we're now taking to try and scale these types of studies up and to make that infrastructure available to people like you. Thank you for your time.
We've got time for a couple questions.
Kaiser database, which is the UK Biobank. How much of that do you think is power? Do you see some of these signatures at lower levels, sort of in the Kaiser database, and is it all gene environment, or is there some element of power?
Right. So we tried to. It's really difficult to nail that down. We can't totally exclude power, but if you look at, say, the ChRNA3 variant in Kaiser, or in males of Kaiser, it looks basically flat. There's not even a hint of anything going on there. Yeah. APOE does show up among mothers of people in the UK Biobank, and the signal in fathers is pretty weak. Unsure why. But yeah, that does partially show up. But the ChRNA3 variant, we've tried poking it a lot in Kaiser, and there's basically nothing there. And also these, the polygenic scores, some of these are whopping signals in the UK Biobank, and we see nothing in Kaiser. And we should, if there was something going on there. At least back of the envelope. We try staring at these types of plots, and there's just nothing going on. But you know, there's different age structures, there's very different analyses, so we can't totally exclude all of that, but we don't think that's what it is.
Question might not be worth getting the microphone for, but did you look at whether that ChRNA3 variant in the fathers extends to impact survival of their wives?
Good question. So yeah, we didn't. Is there sort of a knock-on effect of smoking by a secondhand smoke in the wives of the people in the UK Biobank? And so we basically don't see that signal at all in the mothers of participants in the UK Biobank. So, I mean, it depends, what you expect to see depends on the impact of, you know, the causal effect of secondhand smoke on lung cancer, which is, whatever it is, it's presumably much less than the causal effect of actually smoking. So I don't know if we'd have the power to detect something like that if it existed.
So Joe, in organizing a study like this, do you actually make a list of the genes or the alleles that you expect might be most likely to show up for each of the phenotypes, as opposed to just seeing what the data show? It turns into a sort of hypothesis-driven analysis.
In general here, we've been going entirely agnostic into the genome and not until, for example, we find APOE in Kaiser, then we try to specifically look at it in UK Biobank, for example. But a priori going in, we don't come in with.
It wouldn't change anything you do. It would just add another element of confirming some things instead of always just turning up things to be checked for the additional populations.
Yeah, that's true. That's a good point. So our genetic risk scores are all sort of known variants that we think, well, we want to test whether they have influences on longevity. So those are all, yeah, those are all known variants ahead of time.
In terms of the low-pass coverage for your sequencing versus actual genotyping, what's your accuracy when you actually compare the imputed genotypes from low-pass versus actual genotypes?
Yeah, so we can do imputation. So we do either via simulation or downsampling. You get really low coverage sequencing and then impute it back. And for common variants, the imputation accuracy is around 0.7, 0.6, 0.7. So not great, but for the price, you can't beat it.
Good questions? Yeah.
Do you want to comment on this philosophy that 30x is best and that we should only be doing 30x and nothing lower?
Well, I'm doing 0.1x, so that should sort of answer your question.
If you wanted to get accurate genotypes at an individual level, do you think there's a sweet spot sort of between cost and depth?
Yeah, and I think 30x is overkill for that. I think the data very strongly show that 30x is overkill for that, especially with PCR-free libraries, where you don't have to worry about things as much. Yeah, I think the data I've seen, are more like 20, 22x, 25x for it's basically zero loss in accuracy. If you wanted to then make a slight trade-off in accuracy for cost, you could probably go down to 15, 10x, or if you want to really go the other end, 0.1x.
I think you made a good case on, the more data is a more important than better method, but also methods matter. So in the case of your second project, have you considered you're estimating a lot of parameters that probably lose your power? So have you tried to use a more simpler model like linear model or polynomial model to gain more power? Did you see anything? Did you find anything using them?
Yeah,
those kind of models.
So what we did was in the model I just showed you, there's no form to the relationship between age and allele frequency. It could be totally arbitrary. We did try a model where we insist on it being monotonic, and that gains you a lot of power if the actual effect is monotonic, and loses you a lot of power if there's actually these U-shaped curves or trade-offs. And so for most of what we did, we don't rely on that assumption, but we show it doesn't make that particular. You can do if you force a functional form to that relationship of a linear fashion. In principle, you gain some power, but in practice, it doesn't change much.
Okay, let's thanks, speaker. Thank you, Joe.